So, good morning to all of you students of BT First Sem. We were discussing about the chromosome structure that is given in your cell biology syllabus. So now I am going to continue with the centromere, the other parts which remain in your that I have not described yesterday. So let us describe about the centromere and kinetochore. Well, centromere is the region of the chromosome to which the mitotic spindle fibers are attached, and and the centromere lies within the thinner segment of chromosome called the primary constriction. So, constriction is actually a narrow zone. As you can see here, this portion of the chromosome is narrower. Okay. Um, and that's why we call them as primary constriction. There may be some other narrow regions of the chromosomes as well called as secondary constriction, which I will show you in the next diagram when we will do about the secondary constriction. Okay, so what I have said is about the centromere. Centromere is the region of chromosome where the fibers of mitotic spindle are attached, and these centromere lies within the narrow segment or thinner segment of a chromosome called as primary constriction. So they lie within the region called primary constriction. Okay. And primary constriction are the thin areas or narrow zones of the chromosome, and the regions of the chromosome that surround the centromere consist of highly repetitive DNA. Okay, so within the area of centromere, the DNA that is present around the centromere may contain your highly repetitive DNA. That means repetitive DNA sequence may be present, and they may stain more deeply with the basic dyes. So, as I am saying here, that centromere lies within the primary constriction or thinner segment of the chromosome. Within the centromere, your mitotic spindle fibers are attached so that being able to separate the chromosomes equally to the opposite poles among the daughter cells. And centromere, around the centromere contains highly repetitive DNA sequence, and they stand more deeply with your basic dyes. Now, centromeres are found to contain a specific DNA sequences, and it also consists of specific proteins also which bound to the centromere and form a disc-shaped structure known as kinetochore. Right? Again, I repeat. So, kinetochore, as the name suggests, here in the figure, kinetochore has not been shown. I will try to show you in the next diagram. Kinetochore is actually a disc-shaped structure where the centromeres are interacting with they are actually associating with various special proteins and specific DNA sequence. What type of DNA sequence I have already mentioned, repetitive DNA sequence, and form that disc-shaped structure called as kinetochore. Now, under microscope, especially your electron microscope, this kinetochore appears as a plate or cup-like disc, and the diameter of the kinetochore is about uh, 0.20 nanometer to up to 0.25 nanometer in diameter. So kinetochore appear as a plate or cup-like disc-like structure, and its size is generally of 0.20 to 0.25 nanometer on diameter, and this kinetochore is situated, that is, on the primary constriction or the centromere region. Okay, so let us move to the image where your kinetochore structure has been shown, and interaction of the centromere with the microtubules has been shown properly. Well, here you can see the structure of the chromosome in the above image. You can see that within the centromere region, your the microtubules of the spindle fiber, they are associating during the process of cell division, right? And on the bottom figure, if you see, it shows the kinetochore structure where the proteins has not been mentioned, but you can see that disc shaped structure, which has been shown. And here you can see in the form of noodles, you can write, you can compare them with noodles, just like that. Folded structures of DNA associated with the histone and non-histone proteins are surrounded within this region of within the centromere region, and most of the DNA that is found within the centromere region is of repetitive DNA sequence. Together with that sequence with special proteins, centromere forms a disc-shaped structure called kinetochore. Kinetochore, as I have said, they appear as plate or cup-like or disc-shaped structure whose diameter is about 0.20 to 0.25 nanometer in diameter. And the rod-like structures that you can see right here, these are actually the microtubules which are associating with the centromere portion. Now, during your mitosis, 4 to 40 microtubules of mitotic spindle fiber, they attach to this kinetochore region, as you can see here. You can see here hardly 4 to 5, but it looks like this. You can see here so much microtubules of the spindle fiber are associating. So during your mitosis, you can see the microtubules, about 4 to 40 microtubules of the mitotic spindle become attached to the kinetochore. Here the kinetochore is not been shown, but in the lower diagram you can see the kinetochore structure, and it provides that force for chromosomal movement during anaphase. So the purpose of this mitotic spindle is to separate the chromosomes equally to the opposite poles among the daughter cells, okay, during your cell division. And chromosome already have the I have already discussed with you that your chromosomes have two arms. They have two as a result of replication. You can see, as a result of replication, if I show you in the next diagram, you can see that chromosomes normally becomes in two chromatid forms after the S phase of your cell cycle, and these two chromatids will be separated with the help of these microtubule spindle fibers. Okay, so that equal amount of information of the DNA will be distributed among the daughter cells. Same, actually you can say same amount of, not equal, same amount of DNA information will be distributed among the daughter cells. So here in mitosis, 4 to 40 microtubules of your mitotic spindle attached to the kinetochore, and they provide that force for chromosomal movement during anaphase. And the main function of the kinetochore is to provide a center Okay, it is used to provide a center so that the microtubules can associated with the with that region and separate that chromatids, okay, to the opposite poles of the cell. So again I repeat, the main function of the kinetochore here is to provide the center so that the microtubules can assemble at this site, okay, as shown in the different figure, so that the microtubules can associate in that site. So it provides that platform so that the microtubules can bind for the for that reason. Okay, now you can also say in another way that this kinetochore is serving as the main center for the polymerization of tubulin protein into microtubules. You can say in that way also, as we have known when we are talking about the microtubules. When we are talking about the microtubules are formed by tubulin protein, right? There is alpha tubulin, there is beta tubulin, which forms a heterodimer, and in this way they form these 13 protofilaments to form a microtubule. So in another way we can write about the function of the kinetochore is that my kinetochore is serving as the main center for the polymerization of tubulin into your microtubules, right? Now, most of the chromosomes of most organisms have only uh, one centromere and are known as monocentric chromosomes. Again, I repeat, most of the chromosomes have only one centromere region. That's why we draw in this way, the long arm and the short arm, and in the middle there is a centromere. We give him a round circular like symbol. So most of the chromosomes look like this. Okay, but in some chromosome abnormality, there are some chromosomal abnormality that can be induced by some mutagenic agents which cause change in the base pairs of DNA, which cause change in the structure of the chromosome. Chromosomes may break and they fuse with one another and producing chromosomes with no centromere at all. And such type of chromosome which don't have any centromere are called acrocentric chromosome. Acrocentric chromosome. Again I repeat, as I have said that most of the organisms chromosome have only one centromere. Okay, but there are some cases due to some chromosome abnormality, and that can be induced by using some mutagenic agents like X-rays or by using some chemicals, okay, which can cause the chromosomes to break apart and fuse with one another and produce chromosomes without any centromere, and those chromosomes which don't have any centromere are called acrocentric. Not sorry, not acrocentric, I have said a wrong statement. Acrocentric chromosome is another thing. Acentric chromosome, okay, sorry for that, it is known as acentric chromosome. Acentric chromosome, okay. So acentric chromosome are those chromosomes which don't have any centromere, okay. Due to mutation also there may be a formation of dicentric chromosome as well. As the name suggests, dicentric, di means two, centric, so there will be two centromere which can be found, okay. So this occurs not naturally, this occurs abnormally due to exposure of the chromosomes to the mutagenic agents that causes the chromosomes to break apart abnormally and fuse to form a chromosome without any centromere and chromosomes having two centromere. Now both of these type of unusual structure of chromosome are unstable. Why? Because acentric chromosome don't have any centromere. So how they will be migrated to the daughter cells during cell division? So they will be degraded easily. Okay, while the dicentric chromosomes, um, they will attach to the microtubule. Okay, but due to its two centromeric portions, um, it when they will be trying to pull away from each other, they cause fragmentation. And because since two centromeres found within the dicentric chromosome leads to migration of the chromosomes to the opposite ends, opposite poles, and that's why break of the chromosomes can occur. And due to this breakage, uh, information can be lost, and as a result it is unstable because it has been break from a unexpected site, so it will be degraded easily. So acentric chromosome and dicentric chromosome will not exist for a long time. It will be quickly degraded, they are as they are unstable. Next, the region that I will be talking about is your uh, telomere. So before that, I can say, we can say about the kinetochore function is, if I say about the, if directly if someone asks you what is the function of your kinetochore, then you can say that kinetochore function is to anchor the chromosomes to the mitotic spindle in the spindle apparatus, and it also cause activation of the spindle checkpoint and participation in generation of force to propel, to move the chromosome movement during the cell division. So you can say in this way also about the function of the kinetochore, that kinetochore serve as the nucleation center, that means the main center for polymerization of the tubulin protein to form into microtubules. Then it also, so it is activating the spindle checkpoint, you can say, and they are participating in generation of force to propel, to move the chromosome movement, to move the chromosome during your cell division. Okay, let us now talk about the telomere. Now I have to show the telomere region. For that I have to use a different diagram which shows the telomere. Telomeres are found at the end of the chromosome and they are the protective ends, you can say. So this is the typical structure of chromosome. A single chromosome has been shown. The other chromatid has not been shown. A single chromosome having only one chromatid. That means they have not undergone the S phase. They have not been duplicated. So telomere, as you can see here, if I lengthify the structure of chromosome, it looks like this. This is the structure of a typical chromosome, where you can see a primary constriction has been shown, which consists of the centromere. And here also it associates with different proteins with the DNA sequence that is around the centromere to form kinetochore, where the microtubules are attached, as I have mentioned a little bit ago. But we will now do at the end of the chromosome, which consists of telomere sequence. Telomere sequence are also a little bit of repetitive DNA sequence which protect the ends of the chromosome. It maintains the stability, you can say. And to form the telomere sequence we need a special enzyme called telomerase. We need a special enzyme called telomerase which actually catalyze the that repetitive sequence here, formation of repetitive sequence at the end of the DNA. So telomere is a region of repetitive nucleotide sequence at the ends of the chromosome which protects the end of the chromosome from getting degraded or from preventing the fusion with the other neighboring chromosomes. That's why one chromosome does not fuse with the other chromosomes at the ends, because at the ends it is protected by these telomere repetitive sequences, and these sequences are catalyzed by this telomerase enzyme. You can imagine the telomere as a type of shoelace. When we see a shoelace, at the end of the shoelace you can see there is a plastic coated film you can see, and that plastic coated film can be comparable with the telomere region, just like that. It is protecting the shoelace here, that plastic coated end. Here the telomere is protecting that ends of the chromosome. Okay, so more to go about telomeres. So I have said about telomere regions. Okay, so this is the structure of telomeres, but the sequence has not been mentioned here. Just they have said about the structure. If it was in case of genetics, then I have to show you the structure and how telomere is formed with the help of telomerase enzyme. I have to show, but here that overview structure of chromosomes has been changed, so I am not going for detail here. You will learn them in the genetics class, in your molecular biology class. Then comes another constriction called as secondary constriction region. So let us see about the secondary constriction region. As you can see, if I zoom it out, you can see two type of constriction regions are observed. One type of constriction where the centromere is present, but there may be another constriction as well. Don't mistake with this region of constriction region, because this region is called as secondary constriction region. Secondary constriction, besides primary constriction of the centromere, there may be a secondary constriction region at any point of the chromosome. There is no particular location where that secondary constriction may be present. And secondary constriction are constant in their position. That means they are not mobile, they will not move, and this constriction are useful to identify particular chromosomes in a set. So with the help of secondary constriction, we can able to observe the chromosome within the set of chromosome. We can differentiate one chromosome from the other. Secondary constriction can be distinguished from the primary constriction because the chromosome bends only at the position of centromere during anaphase. So how you can differentiate secondary constriction from the primary constriction is that during anaphase, when the chromosomes are migrating to the opposite poles, you will see that within the primary constriction the chromosome bending occurs. It looks like a this type of L shape, or J shaped, L shaped, or J shaped. Okay, and it occurs on the primary constriction, this bending occurs, or V shaped. Sorry, not like this. Okay, okay, I can draw this. Okay, or V shaped, as I have shown to you in the previous classes, that this type of shape may be observed, particularly in the metacentric, submetacentric chromosome. Acrocentric chromosome may have this type of shapes, and this type of bending occurs on that primary constriction region, not at the secondary constriction region. So you can differentiate secondary constriction from the primary constriction according to the bends that is formed by the primary constriction of the chromosome during the time of cell division, particularly at the anaphase stage, when the chromosomes are getting separated from the uh, opposite mitotic spindle from the opposite poles. Okay, now two more things are left here for completing the structure of chromosomes. One is called the nucleolar organizer region, and another one is called satellites. Well, nucleolar organizer regions are are found within the secondary constriction site. Actually, let us see if it has been mentioned or not. Well, it has been mentioned here, where you can see that one in this chromosome diagram, two secondary constriction has been shown: secondary constriction 1, and on the above secondary constriction 2. Within the secondary constriction 1, you can see nucleolus is interacting with the secondary constriction and forms a nucleolar organizer region. Nucleolar organizer region are certain areas of secondary constriction that contains genes that codes for 5.8S, 18S and 20S ribosomal RNA, and that induce the formation of nucleoli. So this you can say that nucleolar organizer region are the sites of secondary constriction where the genes will encode 5.8S. It will encode 5.8S, then your 18S and your 28S ribosomal RNA. I am repeating here. So this nucleolar organizer region are the sites of secondary constriction where the gene will encode 5.8S, 18S and 28S rRNA. And you know that rRNA are the ribosomal RNA which will further be used for the formation of the ribosomes. Right? Okay. And that's why it induced the formation of nucleoli. As you can see here, nucleolus is associated with the secondary constriction. That's why this region is called nucleolar organizer region. Okay, so the secondary constriction may arise. This type of secondary constriction may arise because the rRNA gene are transcribed very actively here. The genes that are found on this nucleolar organizer region, they code for 5.8S, 18S and 28S rRNA, which will ultimately leads to the formation of ribosomes during translation process. And these are very actively regions, transcriptionally active regions. They are constantly are are synthesizing the rRNA genes by transcription. Okay, and thus they interfere with the chromosome condensation also. So this region are not so much condensed region, because condensed region means what? Condensed regions means the DNA has been coiled with the help of histone and non-histone proteins to form heterochromatin regions. And heterochromatin regions, coiled regions, are transcriptionally inactive regions, whereas euchromatin, or the lightly coiled form of chromatin, they that regions are actually constantly they are transcribing and replicating. So within the nucleolar organizer region, which is located in the secondary constriction, they are all the time as they constantly engaged in transcription of the rRNA genes, like your 5.8S, 18S and 28S, 28S rRNA genes. So that region is normally uncoiled regions. They are euchromatin state each time actually. Okay. Now, in human beings, if I say this type of nucleolar organizer regions are observed in the secondary constriction of the chromosomes like 13, chromosome 14, chromosome 15, chromosome 21, and chromosome 22. So these are the five important chromosomes of humans where you can find this nucleolar organizer region at the secondary constriction. Okay. So what I have said about nucleolar organizer region, nucleolar organizer regions are actually secondary constriction regions, but the main thing is that within this nucleolar organizer region, it contains the genes that are actively engaged in rRNA synthesis. That means constantly they are transcribing the rRNA genes, 5.8S rRNA, 18S rRNA, 28S rRNA, which will be further be processed to form the ribosomal proteins. Okay, and that's why the nucleoli are associating with these regions. Okay, and mostly if I say in case of humans, in humans chromosome number 13, 14, 15, 21, and 22 have this type of nucleolar organizer regions. Okay, so I have mentioned about the nuclear organizer regions. The last region that is, students may get confused of this region also, because it is also found at the end of the chromosome. I have said about the telomere regions also, that telomere regions are also found at the end of the chromosomes. Satellite, it is a bulb like structure, a knob like structure, which called as, which is elongated, round, or sometimes knob like structure, which are found at the end of the chromosome. And satellite remains connected with the rest of the chromosomes by a thin chromatin filament, and chromosomes having satellite are called as sat chromosomes. So satellites are what? Satellites are round, elongated, knob like structure, and they connect with the rest of the chromosomes, as you can see, by a very thin chromatin filament. Satellite is connected to the ends of the chromosome, and those chromosomes that have satellites are called as sat chromosomes. They are called as sat chromosomes. So this chromosome is a sat chromosome. Now, what thing occurs in this satellite actually? What that bulb like structure makes them show? These satellite regions, or the satellite regions of the chromosome, have repetitive heterochromatin or highly coiled DNA sequence that protects the useful euchromatin, that is the loosely coiled DNA, towards the core of the chromosome during replication. So just like telomeres, the telomeres are also protecting the ends of the chromosome. Okay, they're also protecting the ends of the chromosome from getting degraded, or for preventing the fusion with the other neighboring chromosomes. Similarly, here also satellites of the chromosome also consist of a repetitive DNA sequence, and they're highly coiled. That's why we are saying as heterochromatin. Okay, so they consist of heterochromatin regions, highly coiled DNA sequence that protects the useful euchromatin, that is a loosely coiled DNA sequence, towards the core of the chromosomes during your replication process. So this was all about your chromosome structure and functions. With the help of two videos, I have mentioned all of the structures of the chromosomes, also the functions. In the next video lecture, I will try to discuss about the other important organelle of the cell that is given in your cell biology syllabus. Okay, so just go and study these notes that I have provided to you.